Welcome to our video about CRISPR technology and acute myeloid leukemia, also known as AML. We will begin by briefly going over what CRISPR technology is and then go into more detail about AML. We will then spend some time on how CRISPR technology works in order to diagnose and treat AML. Then, we will discuss some of the limitations CRISPR technology possesses as well as some future directions. Essentially, clustered, regularly interspaced, short palindromic repeats, or CRISPR, is a piece of scientific technology that is used to edit genes. Now more widely recognized as CRISPR-Cas9, it allows us to modify, correct, or delete a specific piece of DNA. This is done so through the use of two important pieces, Cas9 and guide RNA. Cas9 is an enzyme that cuts DNA, and guide RNA contains a sequence that directs Cas9 to the target DNA for edits to be made. These two parts form a complex that moves along the strand of DNA until it binds to the matching sequence. Once this is done, Cas9 will cut the DNA at its location, forming a double-strand break, and let repair processes naturally take over. After the cut has been repaired, mutations arise that can have several results. One outcome would be the deletion or addition of several base pairs due to non-homologous end joining. This will in turn interrupt the intended DNA sequence and result in gene inactivation. Large fragments can also be deleted with the use of two different guide RNAs that are placed on either side of the fragment that's to be deleted. Both sides are cleaved and the separate ends are joined through repair processes. We can also correct DNA by adding a DNA template with Cas9 guide RNA complexes that matches the adjacent DNA that's been cut. Using homology-directed repair processes, the template repairs the break in the DNA. Then, the gene is either corrected or a new gene is inserted. Now it's time to introduce you to AML. This disease will be used to further explain the real-life implementation of CRISPR technologies in the real world. AML cancer research is relatively new, with really exciting research developments in terms of technology utilization, which is why we chose to integrate it with the main topic of CRISPR technologies. But first, what is AML? AML is one of the most common types of leukemia in adults. However, AML is fairly rare overall, accounting for only about 1% of all cancers. Acute myeloid leukemia, or AML, is a cancer of the blood and bone marrow. It is the most common type of acute leukemia in adults. This type of cancer usually gets worse quickly if left untreated. But how does AML happen? What's the mechanism? AML develops as the consequence of a series of genetic changes in precursor cells, resulting in an accumulation of large number of abnormal immature myeloid cells in the bone marrow and peripheral blood. Now it's time for some numbers. The American Cancer Society's estimates for AML in the United States for the year of 2022 are around 20,050 new cases of AML with approximately 11,540 deaths. Over the past few years, there's been a lot of work done on improving the diagnoses and treatment options of AML. One of the recent advances in the diagnoses of AML has been with the successful adaptation of CRISPR-Cas9. CRISPR-Cas9 genetic technology has been used to examine the genes and cancer cells of AML mouse models to provide improved treatment options. A recent study conducted by Penn Medicine published an article highlighting their new discovery. They found that the majority of cancer cells in patients with AML relied on a protein called ZMYMD8 protein, and with the help of CRISPR-based screening, they were able to find the exact stop called druggable pocket to target the protein and treat it. Now you may be wondering how CRISPR-Cas9 is used to screen these specific proteins and cancer cells. Firstly, mice models are obtained and AML cell lines are established in these mice. The AML were generated in the mice by transducing leukemia cells into the mouse bone marrow. Secondly, the genome-wide CRISPR-Cas9 screen is performed by using a library that contains over 100,000 RNA proteins and 20,000 protein-coding genes. Thirdly, using previous research and human genotyping of specific genes were matched and screened with the target AML cell lines. Once these genes were matched, they were then utilized and screened in AML mouse models for further evaluation and the survival of the AML cells. Lastly, the genes that survived were then treated with DCPS, a drug that's used to target AML cells. This treatment was then used in the AML mice models to evaluate the successful targeting of genes and inhibition of cancer cells. Although this is an amazing technology, it does come with its limitations. One of the biggest limitations and concerns is that there are frequent off-target effects that are observed at more than 50% of the times it's used. For an example, targeting strand breaking at the unintended location. CRISPR also often induces apoptosis in the gene edit, lowering the chances of success for the intended treatment. Lastly, there is a potential for immunotoxicity that can occur. For example, bacterial infections after using CRISPR may be triggered. Hence, there needs to be more research to ensure its safety. The future also looks bright for CRISPR. In the future, CRISPR may be able to target a large variety of illnesses and is predicted to be the best cancer care due to its versatility and flexibility. Due to technological improvements, there will also be less off-target errors and improve in its effectiveness. There would also be evolutionary changes in the future from CRISPR gene editing, as it will continue to be more affordable and prominent.